Welcome everyone to the Arthritis National Research Foundation's Researcher Spotlight webinar series. I'm Denzel Davis Jennifer, Content Marketing Manager at ANRF. And on behalf of our Board of Directors, Scientific Advisory Board, and staff, I would like to welcome you to the Spotlight on Lupus. We are excited to have so many of you joining us from all around the world. We would also like to recognize our corporate partners for supporting our Research Spotlight series. Thank you to AbbVie for being a gold level sponsor for your continued partnership and dedication. It is because of our partnerships we are able to continue to pave the way for new innovations and learning from our researchers. Thank you. Before we get started, there are a couple of logistical pieces of information we want to share. This webinar is being recorded. We will have it available on our website and YouTube channel in the coming weeks. If you have questions, please type them in the Q&A box. If we aren't able to get to yours, please email them at info@curearthritis.org after the webinar with your question. It is now my honor to turn the mic over to our patient moderator, Adriana Gonzalez, to tell us her story living with lupus.
Hi, everybody. Good afternoon. It's a pleasure to be with you today. My name is Adriana Gonzalez, and I am a 25-year lupus patient and advocate. The path to my diagnosis began at age 11, with my initial symptoms being migraines, chronic fatigue, and pain. 2 years after my initial symptoms began, a kidney biopsy confirmed lupus SLE and lupus nephritis. Living with lupus has had its fair share of challenges that range from extended hospital stays, chronic kidney disease, and a life with daily chronic pain and fatigue. Lupus can be a very isolating, and it can often feel overwhelming to find power in something so difficult. And while I've had to make several adjustments in my life, being able to use my voice to create change and advocate for my community is something that I am extremely grateful for. As a patient advocate for lupus studies, my experience in clinical trials has been extremely beneficial in encouraging other lupus warriors about the importance of participating in clinical trials. That extends to the opportunity of moderating this panel today. Sharing my experience and journey with lupus provides a better understanding of what it's like to live with a debilitating illness. Research and education only brings us closer to a cure. And as a 25-year lupus warrior, I would like nothing more than to see better treatments and a better understanding of lupus. Thank you. I would now like to introduce Dr. Yemil Atisha Fregoso. He is a rheumatologist with clinical experience and experience in clinical research. He has a master's degree focused on study design and analysis and a PhD in molecular medicine. During the last 5 years, he has been studying the role of B cells in patients with systemic lupus erythematosus, designing and performing experiments to study B cell biology. Dr. Atisha Fregoso has published on B cell tolerance in SLE and described a phenotype of antigen-experienced autoreactive B cells in patients with SLE. He is currently working with ANRF to understand the role of incentric and excentric B cells abnormalities in SLE, trying to develop precision therapeutics to improve the prognosis of SLE. Dr. Atisha?
Thank you so much for the kind introduction, and thank you to the ANRF for the invitation to present these results of this project that we are really excited about. So I'm gonna go through what we have now. So first, probably many people here is familiar with this. Like lupus is a chronic disease. Basically, it doesn't have a cure. We can treat the disease, but there is no way actually to get rid of the disease. And the key part of lupus is that it's an autoimmune disease. That means that the immune system that normally takes care of fighting infections attacks the healthy tissue instead, and that causes damage that is associated with a lupus. Anyone can develop lupus, but we know that certain groups, like women, like in reproductive age, are more like susceptible to develop lupus, and also certain like racial groups, like African American, Hispanic people, and other groups also have like a more severe disease. And these have like a hereditary component, which means that people that have a family member with lupus are more have more probabilities to develop lupus at some moment. We don't know exactly what causes lupus. We know that there are certain factors as the UV light produced by the sun, microbiome, smoking, certain medications, viral infections that all together interact with the genes in a person. And this mixture is what we know is associated with developing lupus. What's the exact weight of each factor? We still don't know, but we know that it's like more a multifactorial. That means like many things interact with each other, and that's what causes that some people develop like this disease. One critical component, and this is like basically the focus of most of the research that I'm currently doing and that the laboratory that I have been working, we are focusing, it's autoantibodies. Antibodies are some molecules that are produced by the normal individual in response to an infection. What happens? Like when you have like any kind of infection, like a bacterial infection, a viral infection, there is activation of cells. And this activation of cells finishes with the generation of antibodies. Antibodies are really important because once you have a first exposure to a bacteria, virus, or something like that, the second time that you see the same one, it's going to have like a strong response with these antibodies. And that's going to help to neutralize the infection. That's why when we get vaccines, the idea is to generate antibodies that will protect us against whatever was in the vaccine. These antibodies are produced only by one type of cell that is called plasma cell. However, in autoimmune disease, like lupus, the immune system can produce autoantibodies. These autoantibodies, the auto part means that they're going to attack their own body. Like the same person that's producing their antibodies is going to have like some damage caused by these antibodies. There are multiple types of antibodies. It's beyond what I want to talk here, like to describe that. But there are two types that are important for what I'm going to say. Those are IgM and IgG. These both are like some type of antibodies. In general, in lupus, not talking about any other kind of things like infections. In lupus, IgM is considered to be protective, and IgG is more associated with disease. So I told you before, antibodies are produced only by one type of cell. These cells are called plasma cells, or are the green cells that you can see at the bottom of this plot. So what happened? There are a type of cells that are B cells. These are lymphocytes. For people that is familiar with like a complete blood count, a CBC, sometimes you can see there one value that is your lymphocytes. The B cells, it's one type of lymphocytes. So when there is an antigen, an external link, or in this case, an internal component that causes activation of these cells are going to reproduce. So you have more. Some of them are going to be differentiated, are going to make plasma cells, and others are going to make memory B cells. These memory B cells can be reactivated if they see again whatever causes the first expansion, and they can form plasma cells. In patients with lupus and in certain viral infections, recently it has been described another type of cell, that is one you see here on top, the ABCs, that they follow kind of like a similar pattern that we saw in memory B cells. They are also produced after activation, and they can be differentiated into plasma cells. This is important because we normally don't see these cells in healthy people. We only see these in people that have autoimmune disease or certain viral infections or chronic infections, but they are not normally present in healthy individuals. And the other important concept that I mentioned before, it was that this is caused by autoantibodies. Autoantibodies that attack their own body. But that means that the cell that originally was producing these antibodies, it was able to recognize like self-components, like parts of the body of the person that is making the antibody. Those are called autoreactive B cells. Why we have autoreactive B cells? I mean, that's like a more complex question, but one important point here that I'm not gonna go like through all this slide in general, but what you can see is when B cells are being formed, that they are originally formed in the bone marrow, they have like multiple stages of evolution. During this evolution, every single B cell will develop a specific receptor that is, if an antibody actually, and each one is going to be different. They change the genes in a way that allows them to be able to recognize a different component. Why is this? Because you need to be ready to be exposed to virus, to bacteria. If something like a new virus is developed, you need to have like kind of like a molecule that is able to recognize it. That causes that every B cell have like a different receptor. These are generated mostly in a random fashion. So every cell is going to randomly have a receptor that they allow like to recognize different things. And some of them are going to be autoreactive. When you see the percentage that you see below, it's even 75%, 3/4 of whatever cells are formed are able to recognize self-components. When the cells mature, still 20% of the mature cells that are circulating in a healthy individual are going to be able to recognize self-components. These probably help with many functions, as this can be like getting rid of like some like dead cells or other things. But it's important to understand that healthy people actually have circulating autoreactive cells. So when the mature B cells continue their differentiation until they make like the plasma cells that I described before, fewer and fewer and fewer autoreactive B cells are able to get the next stage. So compared to the mature B cells, only few memory autoreactive B cells are. And basically, almost nothing in healthy individuals reaches the IgG plasma cell compartment. And in patients with lupus, you have like more of these IgG plasma cells that are autoreactive. Okay. So one of the main limitations to study like B cells is that they're hard to identify. However, in our lab, we developed a method that is based on the generation of like we take like some cells that are like some cells that can grow in a plate. We use just a kit to be able to isolate the nucleus of the cell. Then we physically break the nucleus, and we use a molecule that is called biotin. And then we expose the cells from a patient with lupus or healthy individual to these small fragments that come from the nucleus of the cell. Only the cells that are autoreactive are going to be able to identify and bind these things. So you can see how the cell that is binding this is going to be labeled. And when the cell is labeled, we can use a machine that is it's a laser-based machine that is called flow cytometer to identify the cells. So we use this technology, and this kind of a basic process that allows to identify this autoreactive B cell. So this is just an example that how we can see how you see all these cells that are like labeled in the window are what is the autoreactive that we call ANA or antinuclear antigen, because it is coming from the nucleus of the cell, positive cells. So this is what we call ANA positive cells. So with this, and based on what I already described to you, there are many questions that we're interested to understand. So we know that healthy people and patients with lupus have autoreactive B cells. But are these cells more activated in patients with lupus, or what's happening that is causing that patients with lupus are generating antibodies against the self-component or autoreactive antibodies? And is there any target that can help us to attack or deplete these autoreactive B cells that are causing the autoantibodies that at the end are causing the damage in disease? At the end, one of the main goals that we have as rheumatologists is to be able to develop therapies that specifically target like the cells that are associated with disease without affecting another cells. In order to answer that question, we use a combination of the process that I described before, how we label the autoreactive cell, with a new technology that is called single-cell RNA sequencing. This is a high-tech like technology in which we are able to study single cells. So what do we do? So every cell have instructions to establish their functions and whatever they do based on their DNA. These instructions are translated, are like able to make proteins and allow the cell to function using molecules that are called RNA. By looking at this RNA, we can see which genes are being activated or are on in an individual cell. So we can use this technology and we can see the RNA that is present in each individual cell. So what we get is a report card for each cell, what they are doing. They are more activated, they are dying, they are proliferating, what they are doing. So using this combination of cells, we were able to identify and isolate some cells that we defined as autoreactive or ANA positive, as I showed before, and ANA negative in 3 groups of cells. So we included most of the relevant cells that are the precursors of the plasma cells, the cells that will originate the plasma cells. We isolated these cells and we analyzed them using the singles and RNA-seq. So what we got from this analysis, as you can see here in this plot, we use it's a complex mathematical model that puts together all the cells that look similar to each other. So that way we can use a method to see what type of cells have like every person involved in the study of every group of people that was involved in the study. So we were able to identify that there is a group of naive B cells, like these mature B cells, but there is also a group of B cells that are activated. The same for these that is called marginal zone B cells. We found like this group and the activated marginal zone. We found memory B cells and activated memory B cells, and we found these ABCs that I described before. If you see here, this is like already pretty interesting. So you can see how in healthy people, we see basically this pink population at top, that is naive B cells. In the middle, we have like this light purple, that is the marginal zone, and we have some memory B cells. That is kind of what you expected and what we sorted. However, when you see the active patients with lupus, you can see how they have like this big bar that represent the ABCs that are not present in healthy individuals. And for each of the other subsets, they have more of the activated subset than what we can call like the regular or the subset that is not activated. So they have more activated naive, activated marginal zone, and activated memory. We included other group of patients that is also interesting, patients in remission. These are patients with lupus that they have been out of treatment except for hydroxychloroquine at least for 3 years. So this means that they had previously an active disease, but now they are doing well, and they are doing well with the medication. And we want to know how the immune system looks like, because they have been doing so well that they basically don't require medication. But we want to see how activated they are. They are more like the healthy controls, and you can see how they form basically an intermediate state. They don't look like quite similar to the active patients, but they are also not still like the healthy controls. So they still have like some activated cells. And that's especially true for the naive subset. And for the memory, it's basically nothing. So apparently, the remaining alteration is more in the naive and less in the memory. And the other important question, according to what I described, and that's also like really interesting, is, okay, we have the activated cells in the patient that have activity. But in these patients that are having a flare, that are having some manifestation of lupus, how looks like the cells that are ANA positive, the autoreactive B cells, versus the non-autoreactive? And you can see that in this screen, and how they look so different, and how we have all these big group of ABCs that is basically coming from in the ANA positive and not in the ANA negative. And even when you look at the bottom of the graph with the blue and yellow, the proportion of cells that are blue in the ANA negative is 23%, that is a regular memory, and the activated memory is 35%. So it's like pretty similar numbers. But when we see in the ANA positive, you can see that the memory is 9 and the activated memory is 19. So it's less than half. So that means that also in this subset, there is far more activated B cells in the ANA positive. So this means that actually in the patients with lupus that have an active disease, there is some change that is affecting specifically the autoreactive B cells. And now with this information, we can make like some preliminary conclusions. This is still a work in progress, so we don't have the final result yet. But we can say that there are differences in subset distribution between patients with active lupus and healthy donors, that the patients in remission appear to be in a different state that is not equal to the active patients, but they are not like similar to the healthy individuals. That this ABC cluster seems to be like really important within the autoreactive cells. The autoreactive cells that we believe are the one causing disease, the ABC seems to be like predominant in this cluster. And that for the naive and the memory clusters, also more of the ANA positives are inactivated cells. So there is more activation of these cells. And our next step is trying to define exactly which molecules define these activated cells and see if we can develop a target trying to have a specific treatment for only these autoreactive activated B cells. We are really excited about this, and we hope that we will have like results relatively soon that we can share with you like some point. And of course, I mean, I want to say like thank you to so many people. Like making our work like this is like really complicated. It's usually like a teamwork. And so all these people have been working with us, like Dr. Betty Diamond, that is my mentor. I'm working like in her lab. Rita Posovsky is the PhD student that has been working with us, like, and she has been like working really hard in this analysis. All the people that this is like, but also I want to make like a special recognition for all the people that gave us the samples. I know how hard it is for patients with lupus, like all the many times that they need to keep blood for everything, they still get like extra for us to be able to perform this. So we are really grateful about that.
Thank you, Dr. Atisha Fregoso, for that wonderful presentation. This is a reminder for all of our participants on this webinar. Please remember to put your questions in the Q&A box for our question and answer segment later in the webinar. I would now like to introduce Dr. Timothy Niewold. He is the Vice Chair for Research in the HSS Department of Medicine. He is a physician scientist who has pioneered research in underlying genetic causes of lupus, rheumatoid arthritis, and other autoimmune diseases. His dedication to identifying and understanding how autoimmune diseases develops and affects the human body has awarded him many honors, including the Mary Betty Stevens Prize for Lupus Research from the Lupus Foundation of America and the Henry Kungel Young Investigator Award from the American College of Rheumatology. Dr. Niewold has received an ANRF grant in 2008 to study genetic regulations of interferon alpha human lupus, and he currently sits on the ANRF Board of Directors.
Great. Thank you for that introduction, and it's great to be here with you at the ANRF and with all you attending the webinar. Thank you so much for coming. And I'll start with special thanks to ANRF. As you've heard, they had supported me and were one of my early supporters that really helped me get launched down this path. And it started with genetics, and as you can see, I'm still doing genetics. So I just want to give a little bit of an overview of the work that we're doing now to try to help solve lupus. So there's a very nice introduction to lupus in Dr. Atisha Fregoso's talk, so I won't belabor it, but just that as many of you know from personal experience, the lupus can affect many different parts of the body, and it's very difficult in that way. It shows up in many different ways, which can make it difficult to diagnose and also difficult to manage. And every person really has somewhat of their own version of lupus, even though we can unify it under one diagnosis. And this is my sort of summary slide on that. We sometimes call it a prototype autoimmune disease, but I think the manifestations are fairly mysterious or protean, actually. It's multi-organ involvement, and there are some areas that are particularly affected, like skin, joints, and the hematologic and kidneys systems. Very unpredictable. This is very humbling to treat as a physician because you don't know when someone's going to flare up until it happens usually, and it's an unpredictable disease course for most people. And then our current therapies aren't that great. We have immunosuppressants, but they do tend to immunosuppress relatively broadly. Even our new biologic drugs do that. And I'm driven by this last bullet point that we really need a better understanding of what goes wrong in the first place so we can direct our therapeutic efforts in the best way possible. Okay, so how do things go wrong? So this is I'd say it's a little bit of a generic slide, but this applies to many diseases, but definitely to lupus and autoimmune diseases, that there are genetic factors in all of us, and we also have environmental exposures, and these can come together to alter our immune system and then eventually lead to autoimmunity. And so while this slide is fairly simplistic, how it plays out is somewhat complicated. So I'll start off by maybe with the why question. So this may seem counterintuitive that we all have genes that can give us risk of autoimmunity. Many of the genes that have been found as risk factors for lupus may be present in 10 or 20% of the population or more. And there are 100 and some of these genes. So the odds that any one of us has a 0, 1, or 2 lupus risk factors only would be very, very low. Most likely we have a collection of them. So there are a bunch of these genes out there that can give risk for lupus. And that may not make much sense. You think lupus it's not an super uncommon disease, but it's not the most common disease either. It's and so why, if we all have some of these genes, why don't we get it? Well, and where does it come from? So where it comes from, I think this slide explains why. So infectious disease has really been one of the things that has taken us out, basically, killed us through the ages. It's been one of these things where we've been in this almost arms race with viruses and bacteria, and this tends to give rise to natural selection, where we develop genes that are better able to fight off the various infections. And so this is a good thing in fighting off infections, but then we develop this, I would say, immune system hardware through the years that helps us fight these things off. And there's always a chance that any one person might inherit too much of a good thing and actually get too many of these pro-inflammatory genes. And then this could lead to the problem of autoimmunity. And I think that's how it happens for many people, that we have these relatively common risk genes. But if you're unlucky and get too many or the wrong combination, then that could give risk of autoimmunity. Through human history, the risk of dying from autoimmunity versus the risk for dying of infectious diseases has weighed heavily on the side of infectious diseases. Now it's easy to forget that a little more in the Western world because we've got good treatments, although the COVID pandemic is a reminder that this can come back at any time. So I can advance. Okay. And so I said it's complex. So that's how this happens. How can there be this many different common things that come together? They do come together in complex combinations. And I think all of our genetic studies, family studies tend to point in this direction. So for most lupus patients, there may be another relative in the family that have lupus, although many times not. And when we look at large groups of families, of family members who have a lupus patient in the family, there's a greater risk, but it's not enormous. So it's not the same as you would see in a single gene disorder. You could imagine a single gene disorder, either your mom or your dad had it, and then you get it, and then likely one of your children may get it, and it sort of passes down in more of a one-to-one fashion, whereas lupus tends to arise within a family and then wash back out. So the reason for this is that it takes this constellation of genes to come together to really cause lupus. So there are some single gene disorders in rheumatology, and so there are some of these tend to be uncommon diseases like familial Mediterranean fever and other diseases in that family. And there are some rare instances of lupus that are caused by a single gene, but these are fairly uncommon. I would say that this is probably 1 to 2% of people with lupus have a single gene cause, although that's not known. That's just me estimating. It could be more or less than that. Okay, let's see. Advance. Okay. Oh, and how do we detect this? So this maybe give you a sense too. If you look at an individual gene, and say each dot is a person, and you're looking at one gene in everybody, you're trying to see, do you have the lupus risk version of that gene or the non-risk version? And so if you say that the red is a risk and the blue is a non-risk, it'll look something like this. And this would be a strong risk factor, actually, because when you look at this slide, you get a sense, okay, there's definitely more red on the case side. It's usually not quite this strong of a relationship, honestly. Many times it would be hard with a visual plot like this to even tell exactly whether there's a difference. It might be like a 1.3-fold increase on the disease side compared to the case side. Compared to the control side. So there's something there. There's something that's non-chance, and there is something going on. But I think what we're missing is this underlying complexity to be able to know exactly why some people may have this red dot on the healthy side and not have disease, and vice versa. Why do some of the patients, there's no one gene that causes all lupus. So whatever one gene you look at, there will always be some patients that don't have it. So that's kind of the scenario that we're faced with in genetics. I think I've said a lot of this. Probably there are over 100 different risk genes for lupus. Each of them give a risk of disease for about 1.2 to 1.4, and one of the strongest risk factors gives about a 2-fold risk of lupus. And as I mentioned, rare genes can sometimes cause familial single gene cases of lupus, but it's not that common. And I think one of the major frontiers is, now that we've identified all these genes, is thinking about how do they work? This could give us a sense of what really is the human molecular basis of lupus. We know the gene is implicated as a risk factor, and if we could understand how it works, that would lead us to maybe some new insights in the immune system. So we focused a lot of our work in this kind of a thought pattern. Okay. So what are we doing about it, right? That's the scenario. As I mentioned, my work's fairly genetics biased. So each of these 3 things that I'm going to give a short story about has a genetics flavor to it. But we do have one project that's very much in the genes. We call our dark matter project. I'll explain that in another slide or two here. And then there's a specific gene we're interested in studying that we think leads to overactive immunity in your immune cells. And those are some of our major, we heard a lot about B cells just now. T cells are another major cell in the immune system, and dendritic cells, and they all tend to collaborate together in our immune responses. And then one of my most exciting pieces of news lately is that we've actually been able to take one of these gene targets that was discovered in GeneScreen all the way up to a phase 1 trial for an agent in lupus. So I'll save that for the finale, and I'll tell you a little summary about what we're doing there and where that's going. So, okay. So we'll start with this PMP gene. This is sort of maybe one of the simpler ideas, is that we found there's a gene variant in this PNP gene. That's just the name of the gene. The longer name is long. It's purine nucleoside phosphatase. We found it was linked to lupus and interferon, specifically in lupus. So interferon is one of our antiviral molecules, and it's abnormally elevated in many lupus patients. Interferon is more famous now after anifrolumab has been approved as an anti-interferon drug, and is one of the pathways we think is important in disease. About 30% of patients carry this gene variant. We know that the gene variant is less functional, so the lupus-linked gene variant is less functional than the normal gene, and it does impact type 1 interferon and the cell cycle. So right now what we're working on is we've made a mouse model, because this gene is active in many different cells and tissues. And so we're actually going backwards from human to mouse, because there are many studies you can do in mouse tissues and cells that you obviously could not do in humans, to try to understand where this gene might be most impactful. At the same time, we're still doing some human studies as well, and we're studying human immune cells taken from blood or cells that have been cultured in the lab, to understand how this gene works through the cell cycle to impact interferon. So this is exciting. It's one of the examples where we've really nailed down one gene of high interest, and we've even taken it now backwards into the mouse, so we could really get some of the mechanism worked out. Oops, click forward. Okay. Our other genetics project, this is probably the most clearly, this was the one that was first on the slide back there, is the genes project. So we've been interested in the way these genetic associations work, is that we find some regions of the genome that clearly implicate one specific gene, so like PNP on the last slide, or some specific gene structure. But an interesting thing about our human genome is only about 1% of our whole genome is actually the genes that we think of the most, so the genes that encode our proteins and then make up the building blocks of our body. So it's not junk. I mean, it has been called junk. I don't believe it's actually junk. I think a lot of it is there for a reason, and a lot of it regulates the genes. So I think the parts that is not made up by genes is actually doing things to regulate the genes that are there. And so we've taken on this, the project that we call kind of a fun name, Dark Matter Project, is looking at regions of the genome that are clearly associated with lupus, that are risk factors for lupus, that don't contain a gene. And so we focused in on one of these. They're sometimes called gene deserts because they don't have a gene within the region that you could clearly implicate. And it's interesting because in these regions, we often find other non-gene elements like small RNA molecules and or sometimes these things they call longer non-coding RNA molecules that are present in the region. And we think that these are probably being expressed to some degree inside the cell and then modifying some of the other immune responses. So this is exciting. We are hopeful that this may give us some new insights into how lupus works and how the genes impact lupus. Okay. And then I wanted to end with a couple slides about, we'll start with this gene, but this is a gene that we're actually working to intervene with in lupus. So there are gene variants in this ITGAM gene. The immunology name for it is CD11B, which is probably not any less foreign or strange, but it's a surface molecule, meaning it's on the outside of cells, and it can bind to different things in the extracellular space or the outside space from the cell. And it's interesting in that it can turn down toll-like receptor signaling or TLR signaling, which is one of the major inflammatory pathways in lupus. So we found that there were gene variants in this gene that were present in about 20% of lupus patients or lupus nephritis patients. They resulted in a less active molecule, meaning that this molecular brake was less active. So you sort of had less ability to put a brake on inflammation. And then when we worked on these graphs down here, this is just a fancy genetics plot showing that these high dots that are raised up off of the x-axis and ITGAM, those indicate the gene is linked to lupus. And then there's some inflammatory marker graphs over here showing that if you carry the risk gene, whether it's with type 1 interferon or the SUPAR molecule, you had higher levels of inflammatory markers if you carried the ITGAM risk gene. So that's from real, patient data. We've also done some mouse data to sort of back it up and to drive the development of the therapeutic agent. And so with the collaborator at UTMB, Vineet Gupta, he has developed this ONT01 molecule that is an activator of CD11B. So it's trying to turn on this brake of toll-like receptor signaling. And as it shows in the picture here, trying to put the brakes on this inflammatory process here on the picture on the right, where you stop the toll-like receptor signaling and you sort of get the circle with a line through it for inflammatory mediators. So we're very excited about this. It's a small molecule as opposed to a biologic, so the biologics, a lot of the newer drugs have been biologics lately in rheumatology, and those are protein drugs that you usually take through an injection or an infusion. This one is a small molecule, so it's actually a pill-based drug. So our goals in the phase 1 study. Phase 1 is sort of the first study that's done typically, and we had there's one previous study done with this molecule in oncology, and it hadn't been taken forward, so the safety looked pretty good. But usually the first few studies in phase 1 are largely focused on safety to make sure that when you go first into humans that everything is okay and works as you think it will. We'll also explore though whether it impacts some of the molecular features of lupus, and we'll also look at all of the typical clinical measurements in lupus. For those of you who've been in trials or followed trials, there's like the SLEDAI score and the BILAG score, which are acronyms for some of our clinical measures that we use in clinical trials. And the short trial, so in phase 1, you do relatively lower dose with some increase to make sure it's tolerated, and you do it for a pretty short period of time and then see what happens. And then if that all looks good, which we hope it will, we don't know, we'll have to find out. But if it looks okay, then we would then be able to consider moving to a phase 2 trial where you start to look at whether the molecule is actually clearly efficacious in treating lupus. So that is one example I'm very excited about, where you could go from gene and at least into an early phase trial. Again, there are many things that don't make it out of early phase trials, and we don't know yet, but I'm excited to see whether this one might work. Thank you. all for joining today. And again, thanks to ANRF, not only for having me today, but for their support through the years. I've been, it's really a big boost when you're getting your laboratory started to receive one of those early grants. And it was a big deal for me. And yeah, and it's, it's kept me in the lupus genetics world. So thank you.
Thank you so much, Dr. Niewold, for that wonderful presentation. We are going to begin our Q&A shortly, so please, just a reminder, if you have any additional questions, please go ahead and put them in our Q&A box. Okay, I think we'll go ahead and get started. We have a question, and I think this will be both to Dr. Niewold and Dr. Atisha Fregoso. The question is, a lot of time and talent by many researchers have been dedicated to lupus research in the last 4 years. What are each of you encouraged, excited about in lupus research?
Hey, I guess I can start. I mean, it's a great question, and I have to admit I'm biased a little bit, but I am excited about new molecular targets, and that we might actually be getting some more of the immunology figured out in a way that could instruct new therapies. I think we've seen in the last 4 years a good amount of drug development, actually, and I think that's building upon these years of science that came before the last 4 years. But I think it's an exciting time. Whether it's there are many things. I won't list the number of things, but there are many things that are sort of coming along. And I think we've had some new things get approved in the clinic, and I think we may see more as the years go on. So that's one of the things I think is very exciting, is I think we're seeing some of our labors from the past 30 years start to kind of actually lead to some new things that we have in our toolbox for treating lupus.
Yeah, I mean, I totally agree. It's amazing what's happening now. We had like a couple of decades that things were not really moving. Like we had like the same few treatments that were available, and the last like few years, not only we have seen like a couple of treatments that have been approved, there are like so many right now in the pipelines that seems to be like we will have like something new that will work. I don't know if it's gonna be my molecule, like Dr. Niewold's molecule, like you don't know. But like there are like so many options right now that are showing promising results. Seems to be like we will see moving forward this like relatively soon.
Amazing. Okay, our next question. I would say, Dr. Niewold, this is more directed towards you. It's how long will it take before some of these new findings become actual treatments for patients?
Oh, yeah, I guess if we think in the context, I mean, even I could take my last couple slides there where we're in phase 1. This is presuming, again, that it makes each stage. There's a phase 1, a phase 2, and a phase 3. I know for you, those of you who follow trials, you're aware of that. To go even through that, when you have something that's made it all the way up to a phase 1 trial, it's still usually at least, I would say, 7 to 10 years. I mean, maybe my pharma colleagues could correct me on that, but I think it's somewhere in that range to sort of get through the different phases and actually get to FDA and approval. So it's a long road, and it's harder to say if you're in the earlier phases. If you're before phase 1, there's a lot of work just to get something that looks promising enough to take into a phase 1. So I wish it was faster. I think people are wondering if they could make it faster, if somehow the approval processes and trial designs could be streamlined. But it's there's also a very much a due diligence. You want to be careful when you get to this stage. You have to be very careful, so that you're keeping track of side effects and everything else. Making sure that you're bringing something safe along the pipeline. So that's, I hope that's not too much of a downer, but it does take a little while and a lot of investment. But that's the goal, is to get something that's quality, safe, and that could be relied upon at the end of all that work.
Okay, thank you. Our next question: some lupus patients respond well to biologics while others don't. What factors influence how well a patient responds to biologics, and are there ways to predict or improve a person's response to treatments? Dr. Atisha Fregoso, can we start with you on this?
Absolutely, yes. I mean, yes, for sure, like there are some differences that calculate that one patient responds to something that the other doesn't. The truth is, right now, we don't have like a strong tool that we can use, like to see which is exactly the treatment that will work for you. That's still not there. But there's a lot of research going on. Actually, many of the studies with molecular research, as the one that I presented, one of the things that they're trying to identify is like how you can stratify like the heterogeneity that is in lupus to see which patients will benefit. So the short answer is we don't have anything yet, but I think there is like really like nice information and studies going on that I think will help to understand this in a relatively short term.
Thank you. Dr. Niewold, do you have anything to add?
I agree, really. I think that we're making progress. I think it's a little slower than I'd hoped because we have all this great immunology knowledge, and then some of the things that we tried first really didn't pan out as well as we thought. But I think that I agree, we're getting there. I think that this will be something that we'll see in both of our research careers, both myself and Dr. Atisha Fregoso. I think we'll see some of these things actually make it out into practice.
Amazing. Okay, our next question is, this will be for both of you. So we'll start with Dr. Niewold. Do you see any new tools for early diagnosis coming down the line?
I'm hopeful there too. I really am hopeful because probably I'm probably preaching to the choir here, but we don't diagnose early enough, and the tests we have that are clinically approved and used all the time aren't great, like ANA testing and things like this. We use them, they're important, but we're hoping that there can be some other tools that can get there earlier. So I'll say my bias as a geneticist is I think that we will probably get some much more predictive genetic testing and models soon. It's not simple. So I think that's the issue, is that we can't just look at 1 or 2 or even 3 genes and predict. But I think we're working on that next to try to figure out how you might be able to use the genetic knowledge we have to be able to predict better. So I'm optimistic. It's hard to know exactly when, but I'm hopeful that even within my career, we'll see some of these things that might be able to help do an earlier screen. And I think genetics may be one of them.
Thank you. Our next question is, and we'll start with you, Dr. Atisha Fregoso. Are there any co-diagnosis lupus patients should be aware of and looking out for?
Well, actually, that's a pretty complex question, and I think it's a good question actually. But the answer is yes. And the main, probably the most important, other autoimmune disease. We know that there is a phenomenon that is called aggregation. That when patient that has autoimmune disease can have another ones. Many that are common in patients with lupus is rheumatoid arthritis, Sjogren's syndrome, antiphospholipid syndrome. There are many that can be there. But in the same sense that is a complex question, I think this is more about having a good communication with your doctor, with your rheumatologist, and just share your symptoms. I mean, you see something that is unusual, something that you think maybe this wasn't here before, and I'm not sure if it's it or not. You should ask, because if there is one specialty where diagnosis is especially complicated, it's rheumatology. Even when you have a couple of rheumatologists discuss a complicated case, sometimes it's hard to really get in the same line about what's specifically the diagnosis that the patient has. So I think the most critical part here is having this good communication. Any new symptom that you notice, go with your rheumatologist, try to have short communication, email, message, something, and try to see if that's something that you should worry about.
That's great advice. Thank you. For Dr. Niewold, our next question. There's a lot of excitement about using new technologies that look at individual cells to better understand lupus. But how do we overcome the high cost of these tests to make them more accessible?
Great question, and that's true. And I think Dr. Atisha Fregoso talked about this in his talk about using these technologies. And I think probably the realistic answer is these will likely remain research tools for the foreseeable future, but I think that they will provide information to us that we can't see via other methods, and that we could sort of come up with testing that's much less complex and much less costly based upon those findings. I think it'll teach us about the biology in such a way that will then say, okay, knowing this, we could test this other thing, whatever, more simple thing. So I still hold a lot of hope that it will be very informative. I think it's a ways from being done in your regular clinical laboratory that you would send your blood tests to from the clinics, so that's a ways off. But I think that it will be very important even now and in the near future. Maybe, who knows, maybe the medical laboratory, when I'm getting ready to retire, maybe they'll be able to do that, no problem, just knock it out. But I think it's still a little ways off. but it's I think it's very interesting way to study biology, and that it will maybe give us other things that we can test that would be much more amenable to your regular clinical laboratory.
So I would like to get an answer from both of you on this question. So many of us turn to complementary therapies like acupuncture, meditation, or supplements. Is there any research supporting these integrative treatments? Dr. Niewold, we'll start with you.
Yeah, there is some. I mean, it's what I generally say is that I don't think there's been efficacy quite as good as some of our other more classical medications. So you want, and I say that just to emphasize, don't stop your medications to try these, because I think that hasn't been like a home run where you can just stop your lupus with any one of those things. I'll say that I've seen some studies on meditation that I think have been helpful with symptom control, and I think that applies to many, many chronic illnesses, actually. So I think that's something that could be worth trying if you're interested. It's again, none of these is a sort of a magic wand for everybody either, so everybody's body's different, and everybody's set point is different. But I think if you're interested in meditation, you want to try it, it's a very safe thing to do, and it could help with some of the symptoms, and I think maybe some of the symptoms like fatigue and some of the pain that often we don't get at as well with our traditional medications as we'd like to.
Perfect. Thank you. Dr. Atisha Fregoso?
Yeah, I mean, I totally agree. I think that was like a really good answer. And I think like the critical word, one critical word is actually the question. That is like the word complementary. I mean, this is something that will be added on top of your therapy. I don't think it's a good idea. If anyone is thinking about using this as a therapy for lupus as the only therapy, I think that's probably like a really bad idea. We know that. But there are like many things that actually can be actually complicated for patients with lupus. That's was mentioned, like fatigue, like some headache, like problems sleeping. And is with all these therapies actually can be used as a complement to the, let's call like the standard therapy, trying to get like some, to help with those factors. There are few studies that actually have shown that in patients with lupus, there are like others in general population. And sometimes you need to, like to try to understand what work in other things. Let's try here. And again, this is something that usually you need to do with your doctor, because you need to understand sometimes like this is associated with taking a tea, with something, like natural things. And so you always need to have like your doctor ideally aware of all these extra things. But as complementary therapy, I think they can work. And there is some few studies that show that can help with these extra symptoms of the disease.
Thank you. Our next question, and we'll start with you, Dr. Atisha Fregoso. What are some major challenges in implementation for lupus research to clinical practice?
Oof. Well, there are so many actually, but I think that's the reason we're doing here, and that's like I think it's like with like so many people involved. You can start with budget. I mean, the budget that you have like trying to perform all these studies is like limited. Access to enough patients, like even when you have something that is like it's gonna be like a treatment that can be useful, like recruiting patients for clinical trials, it's becoming more and more complicated. Like these studies are like really expensive. You need to go like to multiple centers. Right now, to have a reliable study, you need to cover like a lot of paperwork. Like just the amount of paperwork to start even a small clinical trial, we're talking about like years just putting everything together to start. So I think like there are like so many things there. Probably I will say budget, is critical, like all paperwork associated and recruitment will be the three that I will choose. But it's something that can be done in there, and we are moving on.
Thank you. And then we'll start with Dr. Niewold here. How can patients be a part of research?
That's a great question. So one way is you can definitely ask your doctors, because they may not always bring it up if they're busy, and there may be studies going on. You also can look around many of the medical centers in your area. If they have research going on, they often will have something on their website, so you can sometimes get a sense of what's going on in that way. The study staff also at your clinic and the medical assistants and the people who room patients often will be aware. And then Lupus, there's a fairly good network between some of the foundations, either your local foundations or the national foundations, that try to connect people with research studies in their area, because there has been a push to try to connect the people that want to be involved in research with the researchers. And there are so many different ways to participate. I'll say that for most of the studies that we do, it involves just giving a couple extra tubes of blood when you have your blood drawn. The phase 1 study ended with will be more than that. They'll be involved taking a pill, but there are a lot of different ways you can participate in research too. So if there are ways that you're interested in and ways you're not interested, it's not a one size fits all. So it doesn't even necessarily mean that you have to join like a trial. There are a lot of studies out there, and many of them are actually not clinical trials. They might be studies that I think both me and Dr. Atisha Fregoso may be doing, where we're examining, the immune cells in blood, trying to understand the disease. And so there are many ways. I think if you're interested, both reaching out with your healthcare network, and your healthcare providers, and also local and national foundations, there's really a nice, I think a nice infrastructure provided by that to try to connect people, so that if you're interested, you could find a way.
That's great information. Thank you so much.
As we wrap up, I want to say thank you so much to our fantastic moderator, Adriana. We so appreciate you for being here and sharing your story. Also to our wonderful researchers for sharing their innovative work towards understanding lupus. You can see firsthand the impact ANRF has in supporting these incredible individuals in their quest for a cure. I would like to once again thank our partner, AbbVie, for your support of this webinar. And lastly, thank you to everyone who joined us for this great event. If you've enjoyed this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps. We will be sending our live attendees a survey after the event. Your feedback helps us find direction for future webinars. Please share your comments with us so we can continue providing important and relevant content to you. And please don't forget to sign up for our other Researcher Spotlight webinars. We will be live at the same time on October 30th with osteoarthritis. Thank you all for coming, and please remember to follow us on social media to stay connected.